I'm going to talk about reading and writing the human genome. So the human body contains trillions of cells, and each of these cells carries a copy of the genome, which is like the instruction manual that tells the cells what to do and how to function. Now imagine if you could read and write this instruction manual freely. How would that impact our health? Right now, the way we study the genome and how it impacts the function and malfunction of organs and tissues is that we take millions of cells from an organ, and we study the average genomic profile of all of these cells. But if you think about it, that's kind of like trying to study how a society functions by asking what's the average height of the people, or what's the average age of the people. It doesn't really tell you much about what each individual is doing. And so just as different people have different jobs in society, each cell has a different function in an organ. So rather than looking at an organ as the average of 1 million cells, what we really want to do is to look at them as individuals. What we really want to create is the human cell atlas, which is like a Google Maps that would give you the location, the function, and the role of each individual cell, each of the trillion cells in your body, and tell you what they're doing. To help create this human cell atlas and bring it to fruition, I've designed microfluidic devices, such as the one shown here, to allow us to isolate individual cells so that we can read and analyze their genome individually at the single cell level. But it's not enough to really just be able to read the genome. Let me give you an example of how it can be useful if we can also write the genome. Because sometimes we read the genome and we discover mistakes, like mutations, and we want to be able to write back to it to fix it. So given the example of human papillomavirus, HPV. HPV is a virus that, when it infects your cell, it actually inserts its own genome into yours. It's permanently there to stay. And this can cause mutations and lead to cancer. Currently, there's no cure for HPV. But if you see, 60% of young women and 95% of all men are at risk of HPV infection because they're unvaccinated, and because, and this could lead to cancer. So as the co-founder of Agenovir, what we're doing is trying to cure HPV. Earlier this morning, you heard about genome editing called CRISPR-Cas9. At Agenovir, we're using this technology to specifically target viruses like HPV inside the genome so that we can delete the virus and correct the sequence that it made a mistake to. When we tried this technology on HPV-positive cancer cells, we can see that we are able to kill the cancer cells very well. So as you've heard from me today, reading the genome can help us to map out our biological instruction manual and tell us what's broken with it during disease. Writing to the genome will allow us, give us the ability to fix those mistakes. So we really need to develop technology on both fronts to have innovation, and I'm here to deliver that innovation.